This next topic is extremely important in physiology. It's section 2.3, protein interactions in your book. And the learning objective is 2.3.2, explain the meanings of affinity, specificity, saturation, and competition in protein ligand binding. So affinity, I'll just quickly explain these and we'll go ahead and look at some examples. But affinity is essentially the strength to which binding occurs between, say, a protein and a ligand. A ligand is something that will bind to a protein. We'll come back and talk about that more in a second. Specificity is the specificity of this protein binding, protein and ligand binding. So in other words, you may have a protein that will only bind to a very specific type of ligand, which would be some sort of molecule that it can bind to. And again, we can come back and talk about that more. Saturation is the idea that when you have a protein that has the ability to bind to a ligand, at some point, if you have a lot of ligand, the site where the protein can bind the ligand can become saturated, meaning that as soon as it binds to the ligand, say the ligand unbinds, almost immediately that site will then be occupied by a new ligand. So the site is said to be saturated, and that's another very important idea that we'll come back to and talk about. And then finally, there's the idea of competition. So you could have a protein that binds to a certain ligand, so a certain sort of molecule, and you may have competition for the binding site on the protein between the native ligand, the normal, natural, endogenous chemical, and some sort of exogenous chemical, such as a toxin produced by, a frog or something like that. A poisonous frog might have, in South America, might have some sort of poisonous toxin in its skin. And again, we can talk about these things in more detail, but that's the overview. And these are extremely important concepts in physiology. Okay, quick, on this slide, we're just going to go through some of the functions of proteins, which are extremely important. Again, we'll come back to these ideas over and over again. First of all, proteins make up enzymes. Enzymes are proteins that speed up chemical reactions. We'll go talk about enzymes in great detail. There are also membrane channels. These are essentially form pores through cell membranes that allow certain substances to pass through, such as ions. That's another idea we'll come back to. There are membrane transporters that help move substances across a cell membrane. So these transporters don't create a physical hole through the membrane. What they do is they will bind to a molecule, such as glucose, and then they'll have a conformational change, a shape change, which then moves the glucose from, say, outside of the cell to inside of the cell. There are also proteins that are signaling molecules, hormones. There are many hormones that are actually proteins, or peptide chains. And there are also other types of signaling molecules that are either proteins or peptide chains, polypeptides. The proteins make up receptors. Receptors bind to signaling molecules and initiate a cellular response. They're also binding proteins, so these are extremely important. So these bind and transport molecules. A classic example is hemoglobin, which can bind. It's a protein that can bind to oxygen and help to carry oxygen through your blood. There are also things like low-density lipoproteins. So this is a type of protein that has a lipid covalently bonded to protein. And so it's a way of transporting molecules such as cholesterol. There are also immunoglobulins. These are proteins with an immune system function, such as antibodies. And then there are regulatory proteins. These turn cell processes on and off, such as transcription factors. Let's go into some detail and talk about enzymes next. Enzymes are proteins that speed up chemical reactions. Enzymes bind other molecules through non-covalent interactions. They have a binding site. I'll show a picture of this in a second. I just want to get some of the terminology out there. So a molecule or an ion that binds to another molecule is called a ligand. This comes from ligare, which is to bind or tie. Ligands that bind to enzymes are also called substrates. And the ability of a protein to bind to a certain ligand or group of ligands is called specificity, as I mentioned earlier. And we'll talk about the induced fit model of protein-ligand interaction in a second. So here's a review of enzymes. I assume most of you have probably learned about them in a previous class, but we have a protein, which is the enzyme itself. And in this case, we have two substrate molecules, a green and a red. Now these bind to the what's called the binding site of the enzyme. And these substrates are held in this active site, this active binding site, by weak interactions. By weak interactions, we mean hydrogen bonds, van der Waals forces, and ionic bonds. So these are non-covalent interactions. So this active site can then lower what's called EA or E sub A, which is the activation energy required for a reaction, and this thereby speeds up the reaction. So in other words, these two substrates could potentially come together in, say, an aqueous medium. They could bump into each other and they could react and form the products. But it may just be difficult for that. They may need a lot of energy to do that. They would have to collide with a lot of energy. That would require perhaps a high temperature, high thermal energy for them to sort of crash into each other with enough force to overcome this thing called an activation energy. But what the enzyme does is it lowers the activation energy. It makes it easier for these two substrates to come together and then form the products. Okay, so in step number 4, substrates are converted into products. So we have the blue and the yellow products, and they're released then from the enzyme. And now we have the active site available for two new substrate molecules. So these principles apply to synthesis of energy-rich molecules, synthesis of signaling molecules, protein-protein interactions, protein-DNA interactions, and the transport of solutes across a membrane. So although I'll be talking specifically about enzymes, if we go back a second, some of these same principles would apply to things like membrane transporters. They're going to have a site where the ligand, which is going to be the substance that's going to be transported across the membrane, can bind to non-covalently. So although we're talking about enzymes, many of these principles apply to other types of protein ligand interactions. This is the induced fit, a picture of the induced fit model of protein ligand binding. So in this case, we have two ligands, ligand 1 and ligand 2, and they can both bind to this protein. And the idea is that they're not really exact fit for the shape of these active binding sites. But once they come close enough, they can interact again through hydrogen bonding, other non-covalent interactions such as van der Waals forces and ionic bonds. And what happens is then that the active site actually sort of changes shape a bit, and maybe the ligand does also. And you have this process called an induced fit, in which they now fit together well. And the idea too is with these sorts of reactions is one of the, particularly with this type of reaction, for example, when we have two ligands, the idea is that having the active binding sites close to each other brings the correct bits of these two ligands together if they're going to, say, interact with each other. So that does occur. Sometimes you'll have two ligands, they'll bind to the protein, and then in doing so, they'll then be in position to bind to each other. So that's another type of reaction that an enzyme might do. Again, continuing with this review of enzymes, we have, substrate is another word for ligand when we're talking about enzymes. So we have enzyme plus substrate leads to an enzyme-substrate complex, which can then turn into an enzyme product complex. And then finally, we have the enzyme plus the product, and the product is released. Okay, so sometimes we don't show all the intermediate steps. We just simply show enzyme plus substrate can give you enzyme per product. And note that this is a reversible reaction in most cases. So the reaction can go forward, it can also go backward. And we might show it in an even simpler fashion. We'd say, okay, here are the substrates, here are the products, and here's the enzyme that's involved. So again, to summarize, so enzymes speed up reactions, they regulate reactions, and they're not altered by catalysis.